So we've been talking thus far about ways that B cells are able to make antibody genes, specifically those antibody genes via VDJ recombination. And now we need to think a little bit about what's actually happening in the B cells themselves throughout this process. And so you can see a B cell here that is going to make some antibodies. The way that a B cell is able to both make antibodies as well as make a transmembrane version of this same protein to act as a B cell receptor is shown here. So this is similar to some of the DNA segments that I was showing you last time. So you can see that here we've got the VDJ region that has undergone VDJ recombination. You can see that we're looking at this, we can see there's still a downstream J that is present. We've started transcribing right before the joined VDJ region, but we do still have this, this downstream J is part of our transcript. And we've got the constant region exons. You can also see that there are a couple of different areas after the constant region, including 2 areas where we can have a poly A site. And this transcript can be spliced in 2 different ways. In one, we end up with this pink piece at the 3' end of the RNA. In one, we end up with these green pieces. The pink piece basically just gives us sort of a little end and hydrophobic segment for the antibody, while the green pieces will give us transmembrane regions. And so by alternative splicing of this same mRNA product, we are able to get both a secreted antibody and a B cell receptor that are otherwise entirely identical. And so when I talk about making the B cell receptor, I'm also really talking about that same VDJ recombination process that we saw last time. And so this process of VDJ recombination is happening in our B cells. And we are going to be seeing the details of how B cells work out the timing of this process and regulate this process, which are major parts of the B cells development. That was phrased weirdly, are major parts of B cells development and sort of the developmental process that those B cells go through. And so what we're really talking about here is kind of zooming in on the development of the B cell going from the hematopoietic stem cell to the point of being a B cell that has a B cell receptor and has done VDJ recombination to make both that B cell receptor as well as that antibody. And so we're going to talk a little bit about kind of some of the early parts of this hematopoiesis and then really zoom in on the B cell development stage. So what you can notice here is that the hematopoietic stem cell is going to eventually, it has the option to develop into any of our white blood cells or in fact our red blood cells that we have. These hematopoietic stem cells are found in the bone marrow and are able to basically give us an entire immune system. And so if something happens where your immune cells are going to get killed, like for example, you are getting radiation or chemo, or if you have an immunodeficiency where your hematopoietic stem cells are missing some kind of gene, maybe they're RAG deficient, so you can't make B cells and T cells, we can actually just give you a bone marrow transplant of hematopoietic stem cells from somebody else, and that will be enough to grow you a whole immune system and give you all of the cell types that are shown here. So hematopoietic stem cells are pretty cool. So you can see kind of the locations of hematopoiesis at different times in the life of an organism here. So you can see that we have the hematopoiesis happening first, starting a little bit in the placenta, as well as in the yolk sac, then going to the fetal liver. But right before birth and from birth on, all of our hematopoiesis, so all of those hematopoietic stem cells, and in fact, all of our B cell development will be going on in the bone marrow. So we are completely in the bone marrow for all of this process that we're talking about. And you can see that in a graph on the left, as well as in that figure from your textbook, or sorry, graph on the right, figure from your textbook on the left. So this is an experiment that has been done in order to look at some details of hematopoiesis. And so in this experiment, what people have done is that they have taken some mice and they have given those mice radiation. And they've given those mice enough radiation that it will kill basically all of their white blood cells. And the reason why that happens with radiation or with chemo is that in both cases, you're killing cells as they that are rapidly dividing. They can't repair all of the DNA breaks you're making. And most of your cells of your immune system are pretty rapidly dividing. So when you get radiation, you basically can sort of imagine erasing the entire immune system. And what we can do is we can give that mouse bone marrow as a bone marrow transplant. And when we do that, we restore hematopoiesis and the mouse lives. And so here you can see an example of just sort of giving that mixed bone marrow. So there are a bunch of different cells shown as circles. Each of those cells is sort of different. So you can see you've got L's for some lymphocytes, you've got some eosinophils, you've got some red blood cells as E's, you've got some basophils as B, neutrophils as N, all that good stuff, as well as the stem cell in S. So you can see that in an experiment, we might give a mouse normally about 2 × 10⁵, which is 200,000 cells. One thing that's not shown correctly here is that you actually do this as a tail vein injection. I've done a lot of tail vein injections in the past, and I will tell you that they are rather tricky and rather hard. So if you actually try to give the mouse 200,000 cells, you're probably not going to get all 200,000 of them into the mouse. But people have actually done some further experiments to try to understand what's going on with those hematopoietic stem cells. And so they've done experiments where they actually try to purify those hematopoietic stem cells to see how many stem cells does it take to grow you a whole immune system. And so they have some experiments where they put in 20 hematopoietic stem cells. And when they did that in 4 different mice, 4 out of 4 of those mice, all the mice grew a whole new immune system. And so 20 is enough cells to give you a whole immune system. And they tried 10. When they did 10 out of the 11 mice had a whole immune system. So in most cases, 10 was enough. You can see 5 was enough in some cases, 2 was enough. And in fact, you can actually get some of those mice developing in a new entire set of white blood cells after transplanting in just 1 hematopoietic stem cell. And when I look at these numbers, I say that they are probably kind of overestimates of how many cells it takes because of how hard that injection looks. So my guess, for example, with the 10 cells is the reason why the 1 mouse didn't get a new immune system is not because of the 10 cells not being enough in that case. It's because the injection got messed up. And so my guess is actually in most of these cases, 1 or 2 cells probably would have worked. It just gets really hard to do the injection perfectly when you're injecting 1 cell. So these hematopoietic stem cells are pretty strong and pretty able to actually, give you basically an entire system from that 1 cell type. So that's pretty cool. So as we talk about B cell development, we are going to be thinking about events that are happening in primary lymphoid organs. and specifically in this case in the bone marrow. So remember, we had two primary lymphoid organs, the thymus and the bone marrow. When we're thinking about B cells, we are thinking about the bone marrow. In fact, if we're thinking about the development of any blood cell type except the thymus, we're talking about bone marrow. So thymus is for the T cells, everybody else is in the bone marrow. So right now we're looking at the bone marrow. If you look at this slide and you look at these figures, you can see that there are pretty highly ordered structures in both of our primary lymphoid organs. You can see both in a microscopy image as well as in a drawing from your textbook that there is a fair amount of structure within those organs. And that structure is really important. So you can see that bone marrow structure here. And all of the steps we're going to see today are happening in the bone marrow. So you can see we're in the bone marrow here, where all of the cells are developing. Notice that this is referred to as the antigen independent phase. So at least for right now, think about this as having no antigens. I'm going to tell you a little bit on Monday that that's slightly a lie. But for right now, imagine this as having no antigens. So remember, I told you that VDJ recombination happens in the absence of antigen. We're not getting any input from the antigen through the process we're talking about today. Also notice that at the end of this process, when the cells are being exported out of the bone marrow, there are about 5 million cells that are making it out of the bone marrow as mature B cells every day. So every day of your life, you make 5 million new B cells through this process. So you did that yesterday, you're doing that today, and so on. So nice job. Very impressive. And as I just told you, this process is the antigen independent phase. So we are right here looking at the generation of the diversity of these cells. They are not seeing antigen, they're not expanding. That's all happening later in the response. So we're here at this generation of diversity phase, and we're going to be kind of doing this stem cell to these cells with the receptor on their surface today. So you might ask a little bit about why the bone marrow is important and why this process is happening in the bone marrow. And so you can see two figures talking about the bone marrow here. This is going to get us into an important point about a lot of different cells that we see in the immune system and in their development process. And so this is something that a theme that you're going to see again and again this semester. And so what you can imagine is our developing cells are in fact kind of needy. And so you should imagine them as being kind of needy. Basically, there are some different ways we can think about sort of the psychology of what I'm going to say to you, but we'll just imagine this of the cells. Those cells need a lot of messages that say, you're doing a good job. You should stay alive. So those cells are needing pats on the head or else they're going to die. Basically, if those cells are not getting constant good job signals, then they're going to die. The bone marrow is really important because the bone marrow is a place where there are lots of different types of support cells that are able to provide those signals to the cells as they develop. And so you can see on the top some B cells that are developing, and you can notice that they're sort of shown as green cells, but that there are all of these other cells of the bone marrow that the green developing B cells are interacting with in order to get support and to get signals for that development. You can see something similar at the bottom where our developing B cell, which starts out here kind of brown and becomes that green mature B cell, is going to have lots of interactions with lots of other cell types throughout its developmental process. And that is going to give that B cell the signals that it needs in order to live, which is really important. The one most important signal that I'll mention to you here is a signal that's called IL-7. IL-7 is a cytokine that's made by many of these support cells. It binds to the very creatively named IL-7 receptor on the surface of the B cell, and that is going to keep our B cell surviving until the B cell has a B cell receptor that it can get signals through. So basically, until this cell has a B cell receptor where it can get signals to be told, you did a good job, you should survive, it's going to basically live on IL-7. And that's actually true of all developing lymphocytes. So the bone marrow is really important because it's a great source of IL-7. And so you can see that here as well. IL-7 is going to bind to the IL-7 receptor. We're going to talk more about some specific details of this receptor much later in the semester, and you will suddenly care a lot about this receptor. It's going to undergo some signaling that's going to both allow the B cell to start VDJ recombination, but also to survive and to divide. So it can sort of start going through this whole process. And so this is going to kind of start our B cell on sort of the general phases of its life. So basically, we're going to start following this cell when it has gotten far enough as the hematopoietic stem cell that it has decided that it's going to be a B cell when it grows up. So it's committed to being a B cell, but it hasn't really done anything yet. That's kind of where we're starting with this cell. And it's all in the bone marrow, which is shown in the yellow here. Of course, the bone is in the yellow, before we go later to the secondary lymphoid organs that are in the pink. So before I can give you all of the rest of the information about B cell development, I have to tell you kind of one other piece of general B cell biology. As I mentioned before, for our B cells to develop, they need a lot of pats on the head to get signals to be told that they're doing a good job. And eventually, when the B cell is sort of complete, it is going to get those some signals throughout its life to potentially be activated. And so we do have to think a little bit about what sort of those final signals are going to look like, to think about what kind of signal our B cell eventually needs to be able to get. And so here we're thinking about how we activate a B cell. So this is kind of a very general way that you can understand some aspects of signal transduction that we are going to see again and again throughout this semester. I feel sort of sad that I'm not doing this in person because you aren't going to get to see my interpretive dance of signal transduction. So you're going to have to kind of roll with us here. And so very often when we have receptors on the surface of the cell, very often they have two chains. That's actually true of IL-7 receptor. It's true of many receptors. And so you can think of those two chains as sort of two proteins that are transmembrane proteins that are diffusing around in the cell membrane. One thing that people sometimes forget when they think about transmembrane proteins diffusing in the cell membrane is that those transmembrane proteins are not actually necessarily diffusing together. So one of them might go in one direction, the other might go in the other direction. They're not usually next to each other. They're usually kind of diffusing around all sorts of ways randomly throughout the cell's membrane. That means that normally their extracellular domains are not next to each other. That also means that normally their intracellular domains are not next to each other. If, however, the ligand comes nearby, in the case of the IL-7 receptor, this would be IL-7. In the case of other things, it could be whatever the ligand is. The ligand is going to bind to both parts of the receptor. It's going to sort of catch both parts of the receptor as they diffuse by, and it's going to hold them together. And so now the extracellular parts of that receptor are held in close proximity. That means that the intracellular parts of the receptor, which were previously not next to each other, suddenly are next to each other inside of the cell. You can see that here in this slide. And now these intracellular parts can actually act on each other. They can have enzymatic activity to act on each other. And so you can see that before, these two kinases were not phosphorylating each other, but when they're next to each other, they can put these little red dots of phosphorylations on each other, and that can lead to signaling. This is basically a proximity model of signaling. This is how most types of signaling work. And very often we think about signaling that looks kind of like what is shown at the top of this figure, which is signaling via something called a receptor tyrosine kinase. A receptor tyrosine kinase usually has 3 parts. You can figure out those 3 parts from the name. So a receptor tyrosine kinase has a part that is the receptor, which means it's the part that binds to the ligand. A receptor tyrosine kinase also has a part that is the tyrosine, the tyrosine is a location that could get phosphorylated. So it's these locations that have the potential to be phosphorylated on the domain. And then we also have a part that is the kinase. And a kinase is an enzyme that can do the phosphorylating. So usually a receptor tyrosine kinase has all 3 of those parts. And thus, when we put together, we can use the receptor part to bring everything into close proximity. And then the kinase part can phosphorylate the tyrosine part. There are also some situations where these functions are done by separate proteins that are sort of closely associated, which is being shown on the bottom here. So B cells do most of their signal transduction via something that looks very much like this, but via the B cell receptor. I know it's shocking from the name that you realize B cells use the B cell receptor. So here you can see a good example of a B cell receptor binding to antigen. If you look at this image, you might notice something that is different about the B cell receptor compared to what I showed you with the receptor tyrosine kinase before. And so this image actually has a lot of ways that it's kind of accurate. So if you're looking at this and thinking like, oh, there's something missing, but it's because the image isn't drawn right. No, I actually picked an image that was drawn right to make this point. So if you look at this B cell receptor, is there anything that you notice that sort of is going to cause a problem for the signaling that I showed you on the previous page? All right, I see a hand from Jamie, so I'm going to let Jamie go.
It doesn't have an intracellular region?
Yeah, there's nothing intracellular. Basically, as soon as this crosses the membrane, it just stops. So we've got this fabulous receptor part. In fact, we have to do VDJ recombination to get this fabulous receptor part. But there's no intracellular part. There's no tyrosine and no kinase. So in some ways, it's a good receptor, but in some ways, it's a pretty lousy receptor because it can't do any of that intracellular signaling that I just told you about. In reality, the B cell receptor travels with 2 friends. Those 2 friends are required for the B cell receptor to signal, and they are known as Ig alpha or Ig beta. They also get names with the CD system that I told you about last time. So they are also sometimes known as CD79A and CD79B. And so this protein actually does diffuse through the membrane, staying next to the B cell receptor. The experiments on that are like super cool. We'll talk about some similar ones with T cells later. Those super awesome. But the idea here is that this is actually providing a cytoplasmic tail. And it doesn't have to vary. It actually, every B cell just uses the same partner proteins, Ig alpha and Ig beta. And the point of them is that they actually provide this area inside of the cell called an ITAM, which contains the tyrosine to get phosphorylated. So the T in ITAM actually stands for tyrosine. You're not seeing the kinase in this view. You just have to believe me here that there is also a kinase. Much later in the semester, we will actually see some details about that kinase. But right now, just believe me, there's also some kinase action going on. And that kinase is going to phosphorylate the ITAMs. Also note that to have a B cell receptor and to be able to signal correctly, we need both a heavy chain and a light chain. And B cell receptors will actually signal typically when they are cross-linked and they are induced into proximity with one another. So usually what needs to happen is we have to have an antigen that brings together two B cell receptors. So it's binding to the heavy and light chains of both of those B cell receptors, brings them really close together, and that's going to allow the phosphorylation of Ig alpha and Ig beta and our signal transduction. So this is what it should look like to turn on a B cell, to give a B cell a signal. We are going to, and there are also some other proteins that are going to cooperate in this process that we will think about much more later in the semester. This is sort of where the kinase part comes in. What is actually being shown here is that sometimes complement receptors binding to complement can actually help this activation too. So this is one way where complement is in fact further helping our adaptive immune response. So now we're going to kind of track our B cell through its developmental process. And so we're going to start with a cell that's sort of around here in this process. It has kind of made the decision that it's going down the B cell route instead of the T cell route, but it hasn't actually sort of become a B cell yet. So that hematopoietic stem cell will go through some processes in order to sort of commit to becoming a B cell. It will make some changes in proteins that are on its cell surface, and it will lose the ability to become other cell types. So you can see that it sort of decides first, no, I'm not going to be a myeloid cell. No, I'm not going to be a T cell. No, I'm not going to be an NK cell. And then is just sort of ready to go to become a B cell. And so the first kind of cell that we're going to really think about is a cell that is known as a pro-B cell. When we talk about the different cells in the process of developing today, we are going to think about two really big details about those cells. One of those details is what is on the surface of this cell. That's basically like what kind of receptors are available for this cell to get attention from, for this cell to get its pat on the head from. So what receptors does this cell have? And also we're going to think about what this cell is doing. And so what events this cell is actually doing. And so here we're going to be thinking about the pro-B cell. This part of your textbook divides up the pro-B cell into kind of a couple of different stages. So you can see that there are early and there are late pro-B cells. It should make sense why we have to have these two stages as we talk through the specifics of this. And so first we can think about our pro B cell, and you can see two scribbled on examples of a pro B cell here. We can think about the question of what's on the surface. And particularly when we're thinking about what's on the surface, we can think about what's on the surface of these cells in terms of the B cell receptor, like heavy chains and light chains and the B cell receptor. And this figure is drawn correctly on this front. So if we look at the pro B cell, we in fact look at both versions, all the versions of the pro B cell that are drawn on this slide. What do you notice is on the cell surface as far as the B cell receptor, the heavy chains and the light chains? So what is on the surface here? Any ideas? I heard something. Go for it. Yeah. I think it was Jamie. Yeah.
A CD19 receptor.
So you see CD19. CD19 is actually not part of the heavy or light chain. It's not really part of the B cell receptor. So do you see anything that's part of the B cell receptor? No. So in fact, we think of the pro B cell as having no B cell receptor on its surface. If you were doing flow cytometry to try to find a pro B cell, you would actually look for the cells that are negative for heavy and light chain. The things that the pro B cell are doing kind of make sense in this front. The pro B cell is making the heavy chain. So it can't have it on its surface yet because it hasn't made it yet. So we divide up the two steps of the pro B cell into the step where we're doing recombination of D to J of the heavy chain. So we're putting a D segment with a J. And then the second part is where we're actually putting a V with that DJ. And so that's how we divide up this into two segments. So we make the heavy chain first. We make the heavy chain as a pro B cell. That is the pro B cell's job. The pro B cell is going to first put a D heavy with a J heavy, and then put a V heavy with a DJ heavy. And so that's kind of the job of a pro B cell. And once the pro B cell has done that job, it needs to actually test the heavy chain that it's made. Because it doesn't make a whole lot of sense to go ahead and make a light chain if you did a bad job making a heavy chain. And so our B cell is now going to test the heavy chain to see if the heavy chain that it has made is correct. And so it's going to do that by putting that heavy chain on the surface to see if the heavy chain works, to see if it made a good heavy chain. Once it puts that heavy chain on the surface, it no longer meets the criteria I told you about before of a pro B cell has nothing on the surface. So now it graduates to being something other than a pro B cell. Once the pro B cell has finished doing VDJ recombination of the heavy chain, it's going to translate it and put it on the surface. And now it's not a pro B cell anymore. It's grown up and it gets to be called the next thing. And that next thing is going to test the heavy chain that has been made to see if that heavy chain is a good heavy chain. And so we're going to have this heavy chain on the surface. If you look at that B cell with the heavy chain on the surface, how does this look different than what you saw as with a normal B cell receptor? Anything missing from a normal B cell receptor? Aaron?
The light chain.
The light chain. We've got no light chain. That's actually a really big problem because the heavy chain doesn't actually fold correctly until it has a light chain next to it. So somehow we're trying to test if this is a good heavy chain, but we don't have a light chain to fold next to it. In addition, we want to get a signal through this, but it's not a finished receptor yet. So it's kind of hard to imagine how the heck it's going to get a signal to be told you did a good job. And so in fact, when we're testing that heavy chain, we have to pair it with something else. So we pair that heavy chain with these two other proteins that are known as surrogate light chains. You can kind of think of them as like the fake light chain. They're the substitute. We haven't made a light chain yet, so we can't pair the heavy chain with a light chain, but we need something there in order to help that heavy chain fold correctly and in order to help us test. This is called the surrogate light chain, and the surrogate light chain is made up of two proteins, one called V-pre-B and one called lambda 5. Later on, we will see something called lambda, and just because immunologists apparently are not very creative, we only know the regular alphabet and the Greek alphabet. We reuse some stuff a lot, so lambda and lambda 5 are not the same thing. And so you can see that on this cell, we have our heavy chain with this surrogate light chain. And we call this molecule the pre-BCR, or the pre-B cell receptor. Once a cell has gotten to the point where it puts the pre-B cell receptor on its surface, we no longer call it the pro-B cell anymore. We now call this cell a pre-B cell. And you can see that all of the other signaling components will associate with the pre-B cell, so we can get full signaling via the pre-B cell. So the definition of a pre-B cell is that it now expresses a pre-B cell receptor. Let me check something. And we're going to get signaling through that pre-B cell receptor. If the heavy chain was correct, right now we're assuming the heavy chain was correct. We'll talk in a second about what happens if the heavy chain was not correct, and what the heck I mean by a not correct heavy chain, what can be not correct about it. And all of the events that happen in a pre-B cell are dependent upon the signal that the cell gets through the pre-B cell receptor. This is still a little bit of a problem because remember that the antibody or the B cell receptor binds to antigen with a combination of the heavy chain and the light chain, and we don't have a light chain yet. So the binding is still potentially going to be wacky. We're not going to bind to our normal correct antigen yet. It's okay because we're doing this in the absence of antigen. Instead, the pre-B cell receptor, we have actually learned signals because the surrogate light chain has these weird little tails on it. And those weird little tails actually pull together adjacent B cells or adjacent B cell receptors. And so we've got the pre-B cell receptor that's getting pulled together by having surrogate light chain. So that's how we get that clustering, that proximity of the pre-B cell receptor that is similar to the clustering that we saw with the actual B cell receptor when we really have antigen. So this is how the cell kind of makes it work, even though it does not yet have both a heavy chain and a light chain. And when the pre-B cell receptor signals, a few things are going to happen. So the first thing that's going to happen to that cell is going to get a signal that says, you should survive. Good job, cell. Don't die. The more important part of this is if the cell does not get a signal to survive, then it's going to die. So if a cell does a bad job here, it dies eventually. We'll get there. The next thing that this cell is going to do is it's going to do proliferation. So you can see survival being shown here, and as well as proliferation. You can also see proliferation being shown here. So that's pretty important. And that proliferation is also happening kind of at the same time where we turn off RAG1 and RAG2, because we don't really want to have these proteins that could break DNA around at the same time we're doing cell division, because that's a recipe for disaster if you have DNA breaking proteins around when you're copying the DNA. So we basically get rid of RAG for a little bit and we make lots more copies of the cell. And we're doing this because the cell is basically getting a signal that says, hey, cell, you did a good job. You made a good heavy chain. Let's make multiple copies of you so we can pair that good heavy chain with different light chains. So now we're basically getting a whole bunch of cells with the same heavy chain. They can each make different light chains. So we get that combinatorial diversity that we had before. We also are going to do, we're going to actually turn off production of the surrogate light chain. We don't really need it anymore. We made it, we used it, we're done. So we're going to turn off surrogate light chain production. And we're also going to, and so you can see here sort of that our pre-B cells at first, like I just told you, are turning off RAG. And again, it's because they want to be able to do that proliferation that I told you about. This also leads to one other aspect of this process, and this leads us to something called allelic exclusion. And allelic exclusion is super important. So when we turn off, this is going to seem obvious, but I'm going to make it really clear. When we turn off RAG1 and RAG2, that means we can't do any more VDJ recombination. And so previously we were doing VDJ recombination. We were trying to make a heavy chain. And now we've turned off RAG. We can't do any more VDJ recombination to make the heavy chain. And so what happens with allelic exclusion, you can think of as sort of something that's going on at these immunoglobulin loci. So here you see the immunoglobulin loci. We're specifically acting at the heavy chain here on chromosome 14. But what you should remember is that we have 2 copies of chromosome 14. We have one that came from your mom, and we have one that came from your dad. So in reality, it looks like this. There are 2 copies of chromosome 14. One from mom, one from dad. We have just done VDJ recombination on one of them. So let's imagine that this one, actually no. I wonder how many years of teaching on Zoom you have to have in order to have it work. So let's imagine that the one I drew did VDJ recombination. So now we've got a VDJ that has recombined. If the other chromosome did this rearrangement, it would probably not necessarily pick the same V or the same D or the same J. It probably wouldn't add the same P and N nucleotides. It would actually probably end up encoding a different heavy chain. And then our B cell would have the genetic information to make 2 heavy chains, one from on mom's chromosome, one on dad's chromosome. That would be really, really, really bad. I'm going to find the cell that I want, or the slide I want, because Seriously, self? You did not put that. This is like a super important point, self, and you don't put it in. Oh, there it is. If the B cell made multiple different heavy chains, it made a heavy chain from mom and a heavy chain from dad, we might end up with something that looks like this. You can imagine one parent's heavy chain is in blue, one is in green. We would have a B cell that would have all sorts of mixed up receptors. It would have at least 3 different kinds of receptors that you can see here. The one with mom's heavy chain, 2 moms heavy chain, the one with 2 dads heavy chain, the one of each. Those would all bind different antigens. It's possible that some could bind to foreign antigens and some could bind to self. Like what would happen to this B cell where this bound to this, this one bound to, say, COVID or SARS-CoV-2, and this one bound to insulin. If you got infected with SARS-CoV-2, you'd turn on this B cell, the B cell would be like, sweet, I've got to make my antibodies now. And it could make antibodies against insulin instead of SARS-CoV-2. That would be a disaster, both because you wouldn't fight SARS-CoV-2 and because you would make insulin antibodies. We need our B cells to only make one antibody and one B cell receptor. And this also would be a problem because you wouldn't get very strong binding to that SARS-CoV-2 antigen, which is kind of what they're trying to show in this figure, because you'd only have a small number of receptors on the surface that could bind it. Whereas if all the receptors are the same on the cell's surface, you could get some pretty strong binding. So we don't want this to happen. We want the cell to use one allele and not the other. We want it to leave one allele out, which you could also say is to exclude that allele, which is called allelic exclusion. And so when we actually get a proper signal through the pre-B cell receptor, we turn off RAG1 and RAG2, and this allows us to do allelic exclusion. And so this turns off further heavy chain rearrangement. It's sort of like you can imagine that there is a bit of a race here. Both mom and dad's chromosomes are trying to do heavy chain rearrangement. One of them finishes first. When it finishes, that protein gets put up on the surface of the cell. And if that works, this cell stops rearranging the other one, the slow one. That one doesn't get to finish. Because we got a signal to stop this whole process by turning off RAG. So this is basically making sure that the cell only makes one heavy chain. And so you can see that we basically are going to do the D to J rearrangement on both chromosomes. One chromosome is going to finish doing the V to DJ rearrangement first, and it's going to try to get a signal. It is possible that when that cell tries to make a heavy chain, it does a bad job. It makes a non-functional heavy chain. It makes a heavy chain that doesn't really make a good protein, that doesn't really give a good signal. If that happens, we don't have this signal to stop rearrangement. And so the cell basically gets another try. It gets to finish the second chromosome and try again to make a good receptor. And the cell will then try to maybe test that receptor. If this one, the second try worked, then the cell is good. It gets to keep going. If not, then the cell is never going to get a signal to survive, and that cell is going to die by apoptosis. There are a bunch of reasons why that cell might not make a functional heavy chain, why it might make a mistake during VDJ recombination. You can see that about half of the cells that are developing here don't make it through this process. So half of the B cells that you make that tried to develop are going to fail and are going to die because they mess up. And that's going to seem really inefficient. But this is actually the best scenario because it means that you are going to get a super broad repertoire. You are trying absolutely everything to make antibodies. This is how you get antibodies to everything, because you basically are trying to make the most diverse set of antibodies possible. And that means that sometimes you're going to make something that's trash, and it's not going to work. But we can also think on a sort of molecular biology front about why we might get a non-functional heavy chain. And there's one thing that is the most common problem. And that most common problem comes with the junctional diversity process that we talked about last time with the P and N nucleotides. So if you remember, we are going to add and subtract base pairs with the P and N nucleotides. We can add up to 5 base pairs with the P's. We can add some number of N nucleotides. We can also do some subtracting of nucleotides. But there's one piece of information that is not, that we didn't mention here. And I will say that in the history of my time teaching immunology, I've only had a student ask this question once, where they caught this thing that I was not mentioning. There's the one thing I don't mention, and only one person has ever caught it. So don't feel bad that you didn't catch it last time. When I told you about adding and subtracting nucleotides, did I ever mention the number 3? Did I ever say we add 3 base pairs or we subtract 3 base pairs? Did I say it had to be multiples of 3? Or could it be any number of base pairs that we added and subtracted? Jamie.
You said it could be up to 5.
It can be up to 5 in terms of the P.
So that could shift the reading frame.
Yeah, exactly. So we can actually add and subtract any number of base pairs. It doesn't have to be a multiple of 3. We could add 4. If we add 4, that could actually shift the reading frame. It's possible that we could shift the reading frame and the protein could be something that encodes the V, D, and J regions and then has a stop codon and doesn't have a constant region and doesn't have a transmembrane domain. And so we can anytime we are adding and subtracting is not a multiple of 3 base pairs, we're going to get a frame shift, and we could potentially not even get a full functional protein. And so in some cases, when we do this whole VDJ recombination, we're going to add and subtract base pairs so that we don't get a functional protein anymore. And so all of the cells that do that on the first chromosome are going to then have to rearrange and try the second chromosome so that they can get a signal to live. And any cell that messes up twice is going to die as a result of this process. There are some other ways that there could be problems here, but the frame shift problem is really the first one, or really the major one. And so you can see this process here as well. The cell is going to first kind of do the D of heavy to J of heavy. Then it is going to try to do V to DJ. It might succeed on the first try and get a pre-BCR and then continue down the path. It might fail, but it might succeed the second time, be a good pre-B cell, continue down the path, or it might just completely fail both times and die. Once the cell has actually finished getting a signal here, it is actually going to turn RAG back on. And so now it is going to do the final thing that the pre-B cell has to do. And the final thing that the pre-B cell has to do is make a light chain. And so now we're going to start the VJ rearrangement on the light chain. So we've tested the heavy chain. We got a signal that said you made a good heavy chain. The cell lived, the cell divided. And now the cell, all of these progeny are going to turn RAG back on and start making light chain by doing the VJ rearrangement of the light chain. If the cell is successful and making a light chain, that cell now has both a heavy chain and a light chain to put on its surface. It now gets to be an immature B cell. And it now has that IgM on its surface. It's going to turn off RAG now, and it's going to keep RAG off pretty much forever. You'll see an exception to that a little bit on Monday. But now, so this is kind of if the cell does everything right, and if everything happens perfectly here, this is what happens. So we've got these immature B cells that have a functional B cell receptor. So we can say, yay, good job, you did it. However, it's also possible that the light chain was not great. And so we have to test the light chain. So we're going to get signaling again in this cell. And so when we have a light chain on the surface, we can test it first by giving the cell a signal that says, good job, survive. You are now able to survive. We have that cell divide. So we're going to make more copies of the cell. We turn off RAG and again, do allelic exclusion. So we're now going to do allelic exclusion at the light chain. But we can also imagine situations where this developing B cell fails. And one thing that's sort of unique is that the light chain actually is present in two copies. So you have a light chain kappa gene from your mom and a light chain kappa gene from your dad. You also have a light chain lambda from your mom and a light chain lambda from your dad. There are two different kind of constant regions for the light chain. We don't know, and as far as we know, there's no functional difference here. There's no difference between a kappa antibody and a kappa B cell receptor and a lambda. But what we do know is that every B cell always tries kappa first. So your B cell will try to rearrange kappa. If it rearranges kappa correctly on the first try and gets a signal, then it's done. It's an immature B cell. It gets to go on to Monday's lecture. If it fails, then it tries the other kappa. If it's successful, it's good, it goes to Monday's lecture. If not, it tries again. And now it tries lambda. It tries one of the lambda loci. Either successful or fails. Now it tries the other lambda locus. It can then either be successful or fail. And if it fails after the fourth try to make a light chain, it dies. And so we've got four different things. And so in fact, the only thing that we really know about B cells with kappa versus B cells with lambda is that the B cells with kappa succeeded earlier in their tries than the lambda ones. The lambda ones must have taken longer. They must have failed a couple times before they actually did their recombination. And so now our cell is basically going to turn off RAG. So RAG is off now, and we're kind of pretty much done with VDJ recombination. But there's actually one more thing that has to happen to our immature B cell before it is allowed to leave the bone marrow. We're going to now do one more test on that immature B cell before we let that immature B cell leave. This testing is a testing to see if the immature B cell responds to self-antigens. To make sure that we don't have any self-reactive B cells and to make sure we don't make any autoimmune responses. This testing process, so before we didn't care about antigen. When we did that VDJ recombination, it was all in the absence of antigen. Now, but in fact, in reality, we do make this diverse set of cells, including some that are self-reactive, and then we kill all of the cells that make self-reactive receptors and only let the ones that have non-self-reactive receptors out of the cell. So this is the real way that clonal selection and expansion happens. We make this giant diversity of our different lymphocytes Then we test them against self-antigen, and we purge any that actually respond to self-antigens, only let the ones that respond to non-self antigens out, and let them do their job in the rest of the body. So we will talk about this process, which is known as the central tolerance process, on Monday, of how we actually test B cells to make sure that they're not self-reactive.